Good afternoon, class 12th. Till now, we have completed transcription and post-transcriptional events. So what we have got now is a mature messenger RNA, which is having a tail, a cap, and has been spliced. Now, further, this mRNA, this messenger RNA, leaves the this nuclear membrane and moves all the way to the ribosome, where the synthesis takes place, where the actual protein synthesis or the second part of the protein synthesis, which we call as translation, takes place. So before we start with translation, we need to understand about the machinery involved. So the first machinery which is involved in protein synthesis is of course ribosome. And if you remember what has been taught in class 11th, and in bio also you have studied, that ribosomes are made up of two small subunits. These subunits remain separated in the cytoplasm. Only at the time of protein synthesis they come together and they combine. If you remember the prokaryotic ribosome, the prokaryotic ribosome was 70S. S was the Swedberg unit. Now it was made up of two subunits. One was 30S subunit and the other was 50S subunit. So that altogether was not making up to. The addition of 50 and 30 will be 80. But I told you earlier also, it is not considered to be 80, but it is called to be as 70S, prokaryotic. Now on the eukaryotic one, the eukaryotic ribosomes are of 80S types, and they are also made up of two subunits, 40S and 60S. Remember, it was just a recall. Now these smaller subunit and the larger subunit, they come together and they join at the time of protein synthesis and allow the process of protein synthesis to occur. Okay, so here you can see, when we talk about the ribosome, the smaller subunit has a depression into which the larger one fits, just like blocks. You must have played with blocks in childhood, or maybe playing now also, because it's very interesting. It gives a boost to your mental strength. So blocks, just like blocks, these two small subunit and the large subunit of ribosome fix into each other and form a bigger block, which we call as the ribosome, the complete ribosome. These subunit are associated only when the concentration of Mg2 plus ions is increased. Okay, when there will be low concentration of Mg2 plus ions, then these subunits will remain separated. They only come together and combine when there is enough concentration, when there is a high concentration of magnesium ions in the cytoplasm. So that is also being regulated. At the time of protein synthesis, the magnesium content should be higher. Clear? So now a few things I would like to tell you here about the ribosome. See, when the ribosome combines in this diagram, see here, when the ribosome combine, you can see an A site here and you can see an E site and a P site here. A, P, E, three sites are here. A site is the amino acyl tRNA attachment site. Here the amino acids are attached. Okay, and P site is the central portion. This one, it allows the synthesis of peptide bonds, so it is called as P site. And this side you can see E site. E site is the exit site, from where the empty tRNA exits. If you remember, I will just tell you what actually happens here. The ribosome is having the mRNA attached. mRNA comes. Now transfer RNA comes and reads the codon. Transfer RNA reads the codon. Transfer RNA goes and brings the amino acid, enters the ribosome from the A site along with the amino acid, then moves to the P site, where ribosome forms the peptide bond between two amino acids. Now the empty, when the transfer RNA has passed on the amino acid to the chain at P site, it exits from the ribosome from the E site. Okay, so can you understand the sequence from this side to this side, from right to left here, or maybe left to right? It all depends on the structure, but the sequence remains the same. This amino acid side, then peptidyl side and exit side, from where the empty transfer RNA exits this whole ribosome structure. Clear? So this is it. Now what happens here? There is a point mRNA site also present. mRNA is binding. You can see here mRNA binding site. In the smaller subunit, we have an mRNA binding site where the mRNA comes and joins the ribosome. So this smaller portion of the ribosome holds the mRNA in one position and do not allow it to move, because if it keep on moving, the frame cannot be read properly, the triplets cannot be read properly. So the lower subunit, the smaller subunit will hold the messenger RNA in place, and the larger subunit will allow the transfer RNA to come, read the codons through the anticodons. I will tell you about the structure once more about the tRNA, then it will be more clear. So from A site, what happens? The transfer RNA carrying the amino acid comes. At P site, it transfers it and a peptide bond is formed by the ribosome. Then the empty transfer RNA, which has given the amino acid which it was carrying earlier, has given that amino acid. Now it can exit from the E site. So this whole machinery acts in coordination with each other and allows the chain formation to give rise to a complete new protein every time. Clear? So this structure, as far as this structure is concerned, you need to understand what is a prokaryotic ribosome made up of and what is a eukaryotic ribosome made up of. Sometimes in boards they are asking differentiate between the functions of larger subunit and the smaller subunit also. Although they have not asked, but we may expect that this can also happen because you are studying about the subunits also. So the larger subunit provides A site, P site and E site for handling the transfer RNA, and the smaller subunit helps helps by providing an mRNA binding site. So functions of both these subunit is very important. Now when they will combine, that again you need to remember, these both subunits will come together only when there is a higher concentration of magnesium ions in the proximity in the cytoplasm. Clear? So this structure, the smaller prokaryotic ribosome, you need to understand 70S is made up of 30 and 50. Eukaryotic ribosome, that is 80S, is made up of 40 and 60. The functions of larger subunit and smaller subunit in both the types of RNA ribosomes, that is 80S and 70S, remains the same. Both of them, whether it is prokaryotic ribosome or eukaryotic ribosome, both will have an amino acid attachment site and both will have a P site, both will have an E site for exit of the anti-transfer RNA. Clear? Okay, let us continue further.